All right, so let's see the function. So it's the site that generate energy through the glucose oxidation, okay, during the cellular respiration. So it will oxidize the glucose to become energy. Okay, of course, there's a chapter special for this. Don't worry. So the energy will be released in the form of ATP. Okay, which is what is ATP? Is adenosine triphosphate. Okay, which is this adenosine triphosphate to be used. Why they need to convert it into something? From what we know, energy is nothing, man. Okay, we can't. They don't have a form, right? We can just feel the energy. But how? Why they use this? They change the energy from heat into a form, from through the chemical into a new substance. Mix it means that it makes it solidify, something like that. So it will store inside this adenosine triphosphate. So when they want to use, they will break one of the phosphate, become adenosine diphosphate. Okay, from ATP it becomes ADP. So during the release, the energy, the broken bond will release the energy. So the energy will be used up by the cell or by the body. Okay, but we will go through on, I forget which chapter. I think it's chapter, I think quite front one. We will have a chapter like 3, 4, 5 like that. Okay, so jot this down. This is the function. So the main idea is generate energy. Okay, from sugar, glucose. Okay, not just glucose, fructose also can. All this, but we will go through in more detail on the next chapter. All right, all right. So let's continue. So second one is central. Central is something looks like this. Okay, like a cylinder. See, it's like a cylinder. So okay, it's a small cylinder component. Okay, which exists in pair. Normally comes with pair. It used for the, what you call that? It's made up of complex arrangement of microtubules. Tubules is something like a tube. In organic, means that in biology, a tube-like shape, we call it as a tubules. Okay, so that's okay. So it's it make up of complex arrangement of microtubules and does not exist in plant cell. Okay, this one use it for normally for the mitosis. Mitosis is actually a break, how to say, the breakage of a cell from one cell to become two cells. So they need this to pull it so that it can be tear apart. Normally we use it for the reproduction of cell from 1 to 2, 2 to 4 like that. Okay. Done, huh? All right. So now what's the function? Is to form the spindle fiber. Okay, you need to form the spindle fiber during the cell division in animal cell. Spindle fiber are actually you can like make it like this. Okay, you can understand it like this is your cell. So you need to tear it, right? When you tear, there will be some wrinkles like this, right? When you tear, yeah, there are some wrinkles. These wrinkles are actually the spindle fiber. Looks like this up. Like you when you want to tear something up, not like this is not tear the paper like that. It's like you are trying to gulong the paper and then you tear. When you try to tear when you do it slowly, there will be some wrinkles comes up first, right? So then the wrinkles will pull and then tear the thing. So that wrinkle are actually spindle fiber. Okay, and this spindle fiber is created by central. Means that when you want to pull something, your hand is the one create the power, right? So central is the one who do so. Spindle fiber is the wrinkles to tear it. You can imagine like this.
So center is
It's like our hand, the things that create the force to pull. While spindle fiber is actually the result of the pulling. So you will and this you need to imagine this is a protein. So protein is like your muscle. So when you pull something, it will create the fiber. The fiber will attach the what you call will attach some of the organelle. So when you tear the organelle, like there are two organelle, right? They will pull into both different cells so that it can create a complete cell with complete organelle. So this is how. Can you understand? Uh, okay. All right. Then next, Golgi apparatus. So Golgi apparatus is something like this. It consists of what you call that? Consists of a stack of parallel flattened sac. Okay, they are coated by a single cell membrane. So it's actually the flattened sac. Flattened sac means that sac is actually a small pocket. It's like a small pocket, and then I press it, flatten it. You can imagine, like it's like when you are eating the bun, I open a pocket there, so that is called sac. So when I press it, then flatten. So this is how the Golgi apparatus looks like. Then there was a new membrane added at one end of the Golgi apparatus and the vesicle butt off from the other end. So there will be a what you call a new membrane here. So after I press it, it add a membrane. That membrane is to create so that the vesicle is an even smaller pocket. A big pocket, after I press it, everything will be compressed, correct? So let's say if inside there's a like you have a caramel, for example. When you press it, the caramel will like punch it along. That's called butt off. So you can imagine like inside the sac, there's a big sac. Inside got very vesicle. Vesicle is like a small container, very small container, round shape. Inside got thing. So when you press it, it butt off like that. So this is how. That's why you need a new membrane there at the another end. Why? Because after you open, there's an opening, so you need to close it. We need to add another tape. So that tape is like the membrane. I need to add a membrane there. It's like this is the sac. So I need a membrane to cover this up so that when I press, it will butt off like that. You understand? If no, when I press, only comes out from here, correct? The stack. This is the sac inside, correct? This is how. Done, huh? Okay, so let's continue. So what's the function? So you see, when we say it will create a sac, so the sac acts as a container. So why we need to ballot when we want to ballot something? So we use it to, because we want to, like for example, you buy something and then you ballot it. You put it inside a box. Why? Because you want to. When you buy something and then you put inside a box, in what condition that you need to put inside a box? Why? You want to? Like for example, you want to sell something, so you need to put inside a box so that you can use it to, you can mail to that guy, right? That mail is called transport, correct? And then second, for easy to store, correct? Something like that. So the main idea here is, the main function here is to process, modify, and pack and transport the chemical such as protein, all this. Okay, to transport chemicals. Okay, the main idea is that. So when the thing is butt off, inside the vesicle, it will contain the chemical and also the enzyme. Enzyme is something to create is a medium for biochemical reaction. So that inside the vesicle, while it was transported to the organelle or to the cell, the modification and process will keep on going. It will keep on going then until reach, okay, ngam finish. This is how. So that's why the processes and modify. Packs is like for pack, why we pack it? For transport. So this is how. So for Golgi apparatus, the main function is normally modify and transport the chemical. Okay.在他的面前面对面的，你是嫌他的伤口不够大是不是？ Okay. All right. Now next, plasma membrane. Plasma membrane is actually the protection membrane for the cell. So it means that everything is contained inside the plasma membrane. So it's the outer membrane that surround the entire content of cell. Okay, it's the outer membrane that surrounded the entire content of cell, made up of protein and phospholipid. Okay, it's made out of protein and phospholipid. Phospholipid is phosphorus plus fat. Lipid is actually fat. Okay, so it's thin and elastic and partially permeable. What is called permeable? Permeable is like there's a layer, something can go in and out. You understand? Huh? It's like you know there's one thing, I don't know whether you buy before. It's like it looks like a very jelly-like bubble, round color one with fragrance, with smell normally. That you add water, it will become bigger. You know that one, that condition, we add water, it become bigger. Why? Because the water sits inside it, right? That one is called permeable. So that means that it allows something to pass through it. So for plasma membrane, it's partially permeable. Partially means that not everything can pass through it. Means that partly permeable. Means that some of it can, some of it cannot. Why? Because you need to control, right? Cell, then sometimes they need the nutrient. How they get the nutrient? They get it from outside the environment. So how they know that which kind of nutrient can use, which one I don't want, by using this one. Because partially permeable, it only allows the simple nutrient goes in. A very big size one, it cannot dissolve, it cannot eat. So that's why they need a partially permeable membrane so that it will block out all those they don't want to use, the cell don't want to use. Okay, huh? All right. So jot this down first. Okay, huh? So let's see the function. So like I said, it used to separate the content of cell from the external environment. Okay, why they need to separate? As a protection. Okay, so control the movement of substance. You see, in and out of the cell. This is due to the which characteristic just now? Yes, partially permeable. This is important. You need to like what you call it, print it inside your brain. And then it allows the exchange of nutrient, respiratory gases, and waste material between the cell and their surrounding. Okay? Like, okay, can you understand? Okay, good. So jot this down. Of course, this is also this thing also worth a chapter. Chapter 3, I think. We will have a very detailed one, how the membrane play their role. Okay. Okay. So next, rhizosome. So rhizosome is actually a small spherical sac enclosed in a single membrane. Okay, it controls a hydrolytic cell. What is a hydrolytic enzyme? Hydrolytic enzyme is used for hydrolysis. Hydrolysis is actually a process of breaking off something with one of the product is water. So it means that after I break up something, there will be a one of the product after the breaking is water. So we call hydrolysis. Means that hydrolysis is a breakdown of a very big substance into a smaller substance, and the result, there will be one of the thing after breakup will be water. So that's why hydrolysis. Lysis is breaking down. Hydrolysis means that breaking down involves water. Okay. So they need the enzyme. Enzyme is something to make it faster. Okay, it does not directly involve in the process because before and after the enzyme still maintain the same. but it helps to speed up the process, makes it more efficient. Okay, so jot this down. All right, huh? So it helps to hydrolyze the complex organic molecule. Okay, hydrolyze, like I said, the process of breakdown involve water. Hydro is water. So it breaks down the bacteria and component of damaged cell. Because this bacteria and damaged cell means that the cell already dead. So if you didn't break down it, it will get infected and create something which is not should not be occur inside your body. In short, it might go under mutation and become tumor or cancer. Okay. Okay. All right. So next, nucleus. Okay, it's the largest component in the cell. Okay, it's the largest component in the cell. So it's a spherical, compressed, and enclosed in a nucleus membrane with many pores. Means that many pores means that many lubang, so that the nutrient can go in. Okay. So this nucleus, the nucleus, it contains chromosome, nucleus, and nucleoplasm. Nucleoplasm is like the cytoplasm. It's actually the medium inside the nucleus. Nucleus is actually the, like I said, the center of the nucleus, while chromosome is actually DNA, the other name for DNA. Okay. Okay, jot this down. Okay. So let's see the function. So the function is it control all the cell activities. Okay, so it has the chromosome that contain the deoxyribonucleic acid, the name for DNA, deoxyribonucleic acid, DNA. So what DNA use? DNA will determine the cell characteristic and the metabolic function. Means that everything, every activity, and also the physical appearance, okay, are all determined by DNA. Okay, almost everything, okay, is determined by DNA. So DNA is like the storage. Your computer, you need the system to work, right? The system, you need to store something inside. So DNA is actually the storage. Okay. Okay. All right. So let's see ribosome. So ribosome are actually you see in your rough endoplasmic reticulum, there are a lot of dot thing, right? That I mentioned to you. This is the thing that makes it why we say rough. These dot thing are actually the ribosome. So it's a small, compact, and spherical granule. Granule is also can call as sac, but it's a bit thicker. Sac easily pecah. Granule more the structure more stable. Okay, is to put something inside lah. Okay, granule is like those. There are some how to say. There are some vitamin C. Looks like jelly-like orange in color, right? That one is granule. When you pitch it, it will put out. That one is granule. So it means that is stronger. Okay. If sac is like your ubat, the ubat that you put the inside something looks like a plastic one, the one that we telan, then something looks like a plastic. If you got some water, it will become very sticky one. That one is more like the sac. Okay, so it consists of, it has protein and ribonucleic acid. Why is RNA and DNA almost similar? RNA is actually the storage of, it's like, okay, I want to create a protein A, so my computer, I need to key in the template for the structure computer A, correct? So that template is RNA. It's like it contains all the formula and procedure inside. Okay, it's like when you want to watch, it's like a CD. You want to watch, you know what CD, the old type one DVD. When you want to watch a movie, what kind of movie? You need to put that corresponding DVD, right? So that DVD is like the RNA. Come on. And then the ribosome are present in the surface of rough endoplasmic reticulum or exist freely in the cytoplasm. This thing is sometimes is at the rough endoplasmic reticulum. Sometimes it's within the cytoplasm as well. Okay. Yeah. Okay. Jot this down. 哎，搞不懂了，真的搞不懂。Okay. All right. So let's continue. The functions of ribosome is actually the place for the protein synthesis. Is the site of the protein synthesis. So it's like, okay, when the protein comes in, so what kind of shape you want to come out? Then this is the ribosome. This line here is like the formula of the protein. So it will comes in and then it's like a chop inside, and the new one comes out like that. Not really like that. Actually, the process is a bit different, but you can imagine like that. So the RNA is actually like I said, like the template for the what you call for the structure of protein. So it comes in, or this kind of protein we want to use. So chop, okay, comes out that protein, something like that. Okay. Okay, jot this down. Done. All right. So next, endoplasmic reticulum. No matter it's rough or smooth, the function is the same. So it consists of a system of interconnected folded flattened sacs. Like I said, an endoplasmic reticulum membrane is continuous with the nuclear membrane. So there are two types of endoplasmic reticulum. Okay, the rough endoplasmic reticulum has a ribosome attached to the surface, while smooth endoplasmic reticulum does not have ribosome. Okay, jot down only this two. This upper one only. Okay. All right. So let's see the function. So is the transport system within the cell. Okay. So it provide a wide surface for enzyme attachment and biochemical reaction. Why it need to be like folded? Folded is to increase the exposed surface area. Okay, the air to increase the area that will be connected with the outer environment. Okay, so rough endoplasmic reticulum, it transport protein synthesis by ribosome, while smooth endoplasmic reticulum will synthesis and transport the glycerol and lipid, and also carry out the detoxification and of drugs and metabolic byproduct. This one transport, the rough one transport protein, smooth one transport glycerol and lipid. and also the detoxification. Detoxification means that those toxic that we don't want, it will be carried out by will be transported by this smooth endoplasmic reticulum. Okay, so normally that's why that is why protein we need RNA, correct? The RNA is provided by the nucleus. So that's why you see the rough endoplasmic reticulum normally connected to the nucleus. Smooth endoplasmic reticulum is something that it carries something that we don't want, toxic, all this, right? So it will be nearer to the plasma membrane, so the easier to release. Okay. All right, jot this down. Okay, so now let's continue with the plant cell. Okay, plant cell, it has almost everything in animal cell. Okay, but it has their own several different, how to say, the what I call it has their own organelle, means that the organelle only you will only the plant cell have. Okay, so it's like wait I don't get. So it's like this. Okay, it has almost everything. Okay, but there are several things that which is a bit special one. First, it has a where's the Wako? Okay, it has a Wako. Okay, it has a Wako, and then it has a Wako. It only has cell wall. It has not just the cell membrane, but there is a cover for that, which is the cell wall. Okay, a very special one, and then we have a This touch grain, you don't need, we doesn't really learn that. Should be almost similar, I think. Okay, so I guess only this one. So I will try to cover up those that you doesn't need to jot down. The vacuum membrane no need. Yes, there is a chloroplast as well. Okay, this one no need. Large center no need. And this one touch screen no need. It's not in your syllabus. Yeah, only this. So like for this one, the chloroplast. Chloroplasts are those what you call the green pigment, of plant. Okay, so these three are the difference, the main difference of the what you call of plant cell. Okay, and plant cell does not have central. Just now we have learned animal cell, we got a central, right? The one that tear the thing, right? Plant cell don't have, because the how to say, the cell division of plant cell is a bit different. It will create a new cell wall in the middle and then just potong like that. For plant cell, yes. So that's why it's a bit different. Okay. So you can draw this. Okay, like the smooth endoplasmic reticulum and the rough endoplasmic reticulum, all okay, you just draw accordingly lah. Okay, so there's this one vacuole is actually a storage area, so you just need to draw like a big center like that. Okay, all right, write this down. None, huh? All right, so let's see Wako. So Wako is actually a liquid fuel sac, okay, which that liquid is called cell sap. Cell sap is actually the what you call the nutrient liquid. Okay, that's why when people got sick, I don't know whether you have learned this before in the surviving class. Oh yeah, you guys nowadays doesn't your how to say your uniform doesn't give you guys chance to attend the survival camp. So when you like, for example in desert so when you are lost and you are hungry and thirsty, so when you saw the one with very middle, we call what that one, that plant with cactus, you can potong and then you can drink the liquid comes out from the cactus because that is actually the cell sap, comes with lots of nutrient. Okay, so a Wako is surrounded by the tonal plus membrane. Tonal plus membrane is actually the small one here. You see this one, a very small one, you see. The young plant cell has many small Wako, while the matured one only have a large Wako. So the Wako in animal cell is small. So the cell sap contain the water, organic acid, sugar, amino acid, enzyme, and everything. So basically contain all the nutrient needed by the cell, the plant cell. Okay, because Wako is actually the storage, the store, okay, the warehouse for the plant cell. Animal, actually, we normally just neglect that, but animal cell have. Not all, some of it, yes. Like those cells that need storage, like your muscle cell. Muscle cell, you need to store energy, the ATP, right? So they have small Wako for that. Not all animal cell has Wako. So in your case, we neglect it. That's why I didn't ask you to copy. Okay. Okay. So let's see the function. So first, okay, the water is absorbed into the Wako. Okay, and the plant cell will become the plant cell will become turgid. Turgid is fresh. For example, when you go and buy the vegetables, sometimes they will spray the water, right? After they spray the water from lembe, it will suddenly become a bit looks fresh. We say that one is due to the water go into the Wako. So the Wako will expand, water comes in, right? So expand, expand, the pressure become higher. So the cell wall will protect it first, correct? So when that thing increase so it become turgid. Turgid means that the plant cell with very pressure, high pressure, and then how to say high pressure, it's like your balloon, you know, balloon after I blow, it will become a bit hard, right? Hard to press. That one will be called turgid. That's your car, is it outside? It's okay. Good, jump down first. So in the unicellular animal, the vacuole will contract during osmoregulation, osmosis as attraction. Okay, unicellular animal means that it's a single cell organism. Single cell organism like paramecium, all this. So the vacuole is like there is a pacer inside. Pacer is like a rhythmic, the one that create the rhythm. It's like it will like press, press, press. Why they need to press? Because animal cell don't have cell wall, correct? So when the pressure too high, it will explode. So that's why the vacuole need to contract during the osmoregulation, so that all the water will blow out. Can you understand? To reduce the pressure inside, because cell wall can protect the plant cell, but animal cell don't have cell wall. But plasma membrane will explode. It will break. So that's why it need to like pressing out so that the water will keep pressing out, go out, so it will not explode. Because if it didn't release the water will become bigger and bigger and bigger, right? So the cytoplasm, all this will be pressed to the outside until like balloon, I keep giving the air and finally blown like that. So that's why you need the vacuole to contract. We call this as an osmoregulation. Okay.
The vacuole is to spread the water.
When it contract means that when we try to compress it, the water will go up, right? So the water will be released out of the cell. It's like when you walk what you call that, the sponge with water, after I press, the water will go out from the sponge. This is how.
So the
Fresh. Normally we call it as the plant looks fresh. It's like the chew, you know the not really the bubble gum, you know what's sugus, the gum, the candy. It when you press it, it looks hard, right? At first, that one is called. It's like this is what turgid looks like. So after you press it, it will become really lembut, right? That one is not turgid. This is how. Okay. Okay. Then let me check. I think I will stop here for today. Okay, we still have others. Okay, because we are still a lot. If just one, then no problem. But because still a lot, so we will stop here for today. Okay. All right. So any questions? No, so far not yet. Okay. So see you on next class. Bye bye.